Hello and welcome to the St. Louis University Laboratory Safety and Compliance Training. My name is Steve Rasparsky, and we're going to be talking a little bit about biosafety here at SLU. Here in the biosafety program, our goals are to prevent injury, prevent environmental contamination, and comply with federal, state, and local law. You come to work healthy, and we want you to leave healthy. A biohazard is an agent of biological origin that has the capacity to produce deleterious effect in humans. In other words, it can make you sick. Biosafety is a combination of good work practices, engineering controls, and personal protective equipment designed to protect you from biohazards. There are 4 biosafety levels, 3 of which are observed here at St. Louis University. Biosafety Level 2 is the one most observed here at SLU. Biosafety Level 1 carries with it the least amount of risk, whereas Biosafety Level 4 carries the greatest risk. In Biosafety Level 1, well-characterized agents are worked with. These are not known to cause disease in healthy adults. Work is typically done on the workbench, and standard microbiological practices are followed. Here's an example of a BSL-1 laboratory. Notice that benches and floors are easily decontaminated, and there is no cloth furniture in the laboratory. At BSL-1, a biosafety cabinet is optional. Biosafety Level 2 builds on BSL-1, but includes specific training and handling of the pathogenic agents, restricted access to laboratories, and the use of either a biosafety cabinet or splash shielding on a benchtop during procedures where the biological agent could be aerosolized. Here's an example of a BSL-2 laboratory. Notice it's almost the same as a BSL-1, but with splash shielding and a biosafety cabinet. Also, there are signs on the doors when the agent is being manipulated. Biosafety Level 3 is a large jump from a BSL-2 lab. It builds on BSL-2, but also includes very specialized training in handling of the pathogenic agent. There are also specialized engineering and design features within the laboratory, including restricted access and one-way airflow. In a BSL-3 lab, the biosafety cabinet is mandated. Increased PPE requirements are also observed and often include respiratory protection. In this BSL-3 laboratory, notice the negative airflow, the ante-rooms, and the fact that all work is done in the biosafety cabinet. None of the air in the BSL-3 laboratory is recirculated. All of the airflow enters the suite and is exhausted through the roof. This is an example of a BSL-4 laboratory. Again, we don't have these here at SLU, but it's worth taking a look at. Notice the special decontamination tanks and procedures. Also, there's supplied air to the workers. Only highly pathogenic agents are worked with in these laboratories. OSHA requires that anyone working with human blood, body fluids, tissues, or even well-established human cell lines has awareness training of the dangers inherent in these tissues. These dangers include possible exposure to hepatitis C, hepatitis B, and HIV. Training is offered online through the SLU Online Trainer. To receive access and become enrolled in this training, please contact the Office of Environmental Health and Safety. In addition to the bloodborne pathogen training, we also offer shipping or receiving training for those shipping biological agents. DOT and ICAO regulations require training if you're involved in any part of the transport chain. Contact the Office of Environmental Safety for training. Required shippers training includes general awareness, safety, function-specific, and security awareness. You are required to be aware of the dangerous goods regulations. The next couple of slides include the general awareness training. The following dangerous goods are regulated and require specific packaging and shipping papers. Class 1 for explosives, Class 2 for gases, Class 3 for flammable liquids, Class 4 for flammable solids, Class 5 for oxidizing agents and organic peroxides. Class 6 is divided into Class 6.1 for toxic and 6.2 for infectious substances. Class 6.2 is subdivided into 2 categories. Category A for agents that carry with them a higher risk, and Category B for infectious agents that carry with them a lower risk. When shipping a Category B agent, no shipper's declaration is required. Class 7 includes radioactive substances, Class 8 corrosive, and Class 9 is miscellaneous, which includes dry ice. This is important when shipping a biological agent because often dry ice is used. Dangerous goods are hazardous materials. A hazardous material is a substance or material which has been determined by the Secretary of Transportation to be capable of posing an unreasonable risk to health, safety, and property when transported in commerce. Awareness of security risks is required. Hazardous materials can be a threat to homeland security if used illegitimately. Dangerous goods readied for shipment are a security risk and must be secured from unauthorized access prior to shipment. All visitors must be escorted in areas where packaged dangerous goods are awaiting shipment. Securing dangerous goods means keeping them behind closed and locked doors in the laboratory, limiting access to only those who need it, and calling public safety at 977-3000 in the event of any security issue. Here at SLU, we have several entities that provide oversight for experiments. The Institutional Biosafety Committee provides oversight for any experiments involving biological materials. The RAD Safety Committee provides oversight for any experiment involving radiation. For any experiment involving animals, the Institutional Animal Care and Use Committee will review the protocol. And the IRB takes care of any experimentation involving humans. We recommend contacting our office to let us know what kind of research you're interested in doing, and we'll help you determine what category it falls into. Any DNA work must be registered with the NIH. Anything you generate as a recombinant molecule in your lab, the NIH wants to know what you've created. Point mutations in protein genes are supposed to be reported to the NIH. If you know that you are creating something dangerous, or with the potential to be dangerous, those must be registered with the NIH through our office, and through the IBC. Some recombinant DNA work is exempted from the guidelines. All such research must be conducted using BSL-1 practices. Experiments requiring prior approval include gene transfer experiments in humans, genes for toxins that are lethal for vertebrates, release of genetically engineered organisms to the environment, those using human or animal pathogens as host vector systems, including adenovirus vectors and murine retroviruses that infect human cells, cloning DNA from human or animal pathogens biosafety level 2 and higher into a non-pathogen host vector system, cultures of more than 10 liters, and experiments involving whole plants or animals, including the creation of transgenic organisms. Please note that all experiments require IBC approval. Contact the Office of Environmental Health and Safety for more information. And for more information on the NIH guidelines, please visit their website here. The NIH comes around to do inspections occasionally and reviews IBC minutes to be sure that SLU is in compliance with the NIH guidelines. The training you're in here today is a part of the laboratory safety training and includes general, occupational, biological, and chemical safety. Additional training is available for different topics, including SHARPS training, bloodborne pathogens, RAD safety orientation, animal use orientation, and ABSL-3 animal proficiency training. Note that depending on your job description, some of these may be mandatory. The Occupational Health Program Awareness and Enrollment. This is a medical surveillance program for ensuring that employees are monitored for occasional exposures to hazardous materials. The employee's PI will be provided with a questionnaire to complete. For work with animals, the PI will also be provided a safe handling of animals for OHP form. If you choose not to participate in the OHP, you must notify the SLU employee health physician in writing using the Informed Consent Declination document. Note that the Occupational Health Program is very important in protecting you, and if there are any questions, please discuss this with your supervisor. In this next section, we'll be talking about waste disposal, SHARPS handling, labels and signage, PPE, and decontamination. Here at SLU, there are two ways of getting rid of biological waste. Infectious waste that's been autoclaved and placed into a black trash bag can go into the regular trash. The autoclave must be validated using a spore challenge. This is to be completed weekly, and documentation is necessary. All sharps must go into an approved sharps container. These approved sharps containers are puncture and leak resistant. Note that occupants of tenant-owned buildings adhere to their own policies. The other way to dispose of biological waste at SLU is through a third-party vendor called Stericycle. Using this technique, the laboratory has a cardboard box with a red bag liner. This red bag liner meets minimum dart and tear test result and should be the only bag used in the box. These biohazard boxes are specification packaging with appropriate labels. To close the biohazard box correctly, the bag is to be tied with a single square knot. Tying the bag correctly ensures that it's going to be leak-proof if turned upside down for 5 minutes. This is a DOT requirement. Once the bag is tied, the box should be sealed with 3 strands of tape. Here's an example of the bag being tied correctly. The barcode should be placed either on top of the box or else on the side of the box over the words "Customer Label Here." If this sheet is found on a box, please direct your attention to the checked line item. It probably means there was a problem with the bag, and pickup might have to wait another week. The top picture is an example of a sharps container that is puncture and leak resistant. The far bottom picture is an example of a biohazard box that is way too full. Tying the proper knot would be impossible. This picture is an example of a sharps container with a needle coming out of it. Please note that puncture resistant does not mean puncture proof. Here's an example of items that go into a sharps box: scalpels, needles, and razor blades. Broken glass, not contaminated with biological, chemical, or radioactive material, can simply be placed in your broken glass box in the laboratory. Alternatively, any cardboard box could be used. Once full, simply write "broken glass" for disposal, tape the box up completely, and housekeeping can take it. Here's an example of a typical glass disposal box. Any plastic items not contaminated with biological, chemical, or radioactive material can be placed in the trash. OSHA requires biohazard labels to be affixed to containers, refrigerators, or freezers containing human bloodborne pathogens. This is also a CDC requirement. Personal protective equipment in your laboratory is required. However, it varies depending on what you're working with. Disposable gloves, lab coat, and eye protection are almost always appropriate in every laboratory. For instance, if you're working with something and it aerosolizes, eye protection may be the only thing between you and exposure. Most of these substances are acceptable for surface decontamination. Note that ethyl alcohol has a very limited contact time, and alcohol alone may not be suitable to kill the agent with which you're working. It may be more appropriate to use a 1:9 dilution of bleach with respect to bleach, and then go over your surface with alcohol. Phenolics, including Lysol, are not to be used as they have very poor warning properties. Upon direct contact with the skin, they numb the skin, preventing the user from realizing that damage is occurring. Let's talk a little bit about equipment. I've spoken briefly about aerosol generation. There are several ways that a biological material may become aerosolized. Overpressurized needle containers, fluid expression pipettes, and vortexing are 3 ways. It's best to use a biological safety cabinet for any procedures where aerosolization may occur. Your biological safety cabinet must be inspected annually by an outside vendor. There are several classes of biosafety cabinets. Class 2 cabinets are used here at SLU very often. Class 2s provide personal protection from inward airflow. Type A1 cabinets are not suitable for work with volatile or toxic chemicals or volatile radionuclides. Type A2 cabinets can be used with minute quantities of volatile toxins. Please use common sense in the laboratory. Rules are there to minimize risk and to keep you safe. There are certain hazards that come with centrifuges. Mechanical and lab equipment failure is one hazard, but also aerosol generation. Any liquid at the bottom of the centrifuge can become aerosolized upon the operation of that centrifuge, so just make sure that the centrifuge is cleaned up before use. Let's talk a little bit about emergency response now. Small spills can be cleaned and disinfected by laboratory staff. In the case of larger spills, or if you are uncertain about what the material spilled is, please call the Office of Environmental Health and Safety, as we provide 24-hour emergency response. Alternatively, public safety can be contacted at 73000, and the appropriate staff in our office will be contacted. For exposure or contamination with a biological material, take the following precautions. Don't leave the area. Notify other laboratory personnel, including your supervisor, and then call public safety. Again, they'll page the appropriate staff and will come to help. For any sharps injuries, including sharps injuries with a biological material, notify your supervisor immediately. Next, report immediately to employee health. An incident report will be filed, and the biosafety officer will be contacted. There are numerous security measures in place for work with select agents and toxins. These include an FBI background check, fingerprinting, and Department of Justice approval. There is work with select agents and toxins here at SLU. The CDC states that any institution working with select agents or toxins must have one person to whom they can go for any questions or comments regarding select agents or toxins. This person is called the RO, or Responsible Official. The responsible official here at SLU is Mark Campbell. There are two alternate responsible officials, or AROs, here at SLU. They are Steve Rosparski and Mark Henshin. Please direct any questions about select agents to any of these three people. The Environmental Safety website contains a lot of additional information. For any questions, comments, forms, or emails, please visit oess.slu.edu. Here is some contact information from Mark Campbell and Steve Rosparski. This concludes the biological safety portion of our training. Thank you very much for your commitment to laboratory safety, and have a great day.